Hello, today we are going to discuss on differential leukocyte count DLC. Last 3 days you have made all your attempts to do it, focus it under microscope, and after taking the good blood smear and staining it. So in the examination, either in internal or in final examination, they will ask you about the complementing questions on the theory, in addition to your performance with the skill in terms of taking blood smear, as well as in terms of how you focus and project one or two cells. There are two possibilities. Examiner may ask you to project at least one cell and put it under observation. Otherwise, anyhow you know how to make the counts. But the questions complementing it will be based on the theoretical component, like the name of the stain. I think you know the name of the stain. Then you need to put the you need to put the constituents, or I can say what are the components of the stain and function of each component. Once you know the function of each component, they will ask you why oil immersion objective, what is the oil you are using, what is the refractive index of the glass or refractive index of the oil, so that you need to answer all these questions. After this, they may jump to the next level, that is neutropenia, neutrophilia, or lymphocytosis, lymphocytopenia. And the other possibility is how do you discriminate between large lymphocyte and monocyte. Both are agranulocytes. The difference is the size. How do you measure size under microscope? The size measurement is only by calibrating with the size of the RBC. So if 2 times the size of the RBC, that is large lymphocyte, 2 times to 3 times, whereas 3 times to 4 times is monocyte. One more major difference, crucial criteria for identification is the size of the nucleus. Nucleus to cytoplasmic ratio. Nucleus to cytoplasmic ratio is 80 to 20 in case of large lymphocyte. In case of monocyte is 50-50. That means nucleus occupies 50% of the cell, remaining 50% is the cytoplasm. After that, they may ask you all around the clinical applications like eosinophilia. What are the conditions in which eosinophil count will increase? I think these are the 3 examples. It was covered in the theory class also. You need to answer allergic manifestations or allergic situations or conditions, followed by worm infestation is important in India, then skin conditions. So you need to look around all these conditions to get good marks in DLC, differential count, which is a 10-mark question, major experiment in blood. Right, see you.